years as assistant professor at Harvard. Dr. Snyder was recruited to Stanford Burnham Medical Discovery Institute as professor and serves as the founding director of the program in stem cell and regenerative biology, the Stem Cell Research Center, and the Stem Cell Core. His research focuses on stem cell biology. He truly has been at the first mitotic event for many of the discoveries in this field, as he's reported in numerous high-impact publications. Dr. Snyder serves on long lists of scientific and medical advisory boards, NIH study sections, journal editorial boards, has been member of many scientific honorific societies, delivered numerous named lectures. He is a Grass Lecturer for the Society of Neurosciences, made the Forbes magazine list of 12 stem cell revolutionaries, honored by the San Diego Chamber of Commerce, appointed chair of the FDA Cellular Tissue and Gene Therapies Advisory Committee, the NIGMS Human Genetic Cell Repository, and was named Rock Star of Innovation by Connect San Diego. But perhaps most impressively, Dr. Snyder's CV lists 53 current or former trainees, from high school students to visiting professors. One of his former trainees is our own department's Dr. Brian Tobey, who, as we speak, is hard at work running the psychiatric emergency clinic at the VA hospital. In his email to me, Dr. Snyder credited Brian with being pivotal in spearheading much of the work he'll discuss today. Please join me in welcoming Dr. Evan Snyder, whose grand rounds this morning is entitled Using Stem Cells to Model Psychiatric and Other Complex Neurologic Disorders.
Not only is it an honor to be here, I have to say that this is one of the most I'm probably more nervous for this talk than maybe all the others I've given. This will be only the second time that I will have given this story in front of real psychiatrists. And now that's not to say, you know, as I was pointing out at lunch yesterday, technically I am part of the club. If you notice, the boards do start off like that. However, having said that, you know, as Neil said, I was really, really fortunate to be able to have Brian Tobey in the lab, who really spearheaded much of the work that I'll be talking today and absolutely kept me honest whenever it came to that. So if I say something that's outlandish, it's not Brian's fault, it's my own. And Brian was able to recruit a whole host of other well-known psychiatrists, including some from right from here and from this department. Now, as a result, at lunch yesterday, it was mentioned that that I might be the first one to actually kind of give a stem cell related talk here. I don't know if that's true or not, but it was mentioned that maybe I should also give just a very quick little introduction to stem cells and the notion of stem cell modeling before I actually launch into that. And since I have an hour, I thought, and I'd luxuriate a little bit in this because I would give it to you from my particular perspective. Now, everybody's heard about stem cells, and what I want you to do is just listen to what I have to say about stem cells. But what stem cell biology did, and I'm not talking about the therapy or anything else, it just changed our thinking about how the whole body, but particularly an organ that was thought to be absolutely rigid, was put together. It made us think not the way we thought that this was a very deterministic system, but that there was plasticity. And the pendulum swings back and forth as to how much plasticity there is and how to harness it, but we do accept that there is some level of plasticity. Some of that plasticity is attributable to the presence of a stem cell all throughout life, not just early during embryogenesis. So first and foremost, a stem cell is a part of developmental biology. Its role is to put the system together and then to maintain homeostasis all throughout life. And if you go counter to what the biologic imperatives are, you will go awry. So what you need to do, if you're interested in this, is be a first-class developmental biologist. What that means is that if we're going to be talking about the nervous system, and the building block of the nervous system is the neural stem cell, it doesn't matter how you get there, whether you do it from a fetus or an adult or ES cells or iPS cells. You need to know what's the biologic language that cell is talking. what it is meant to do, as I mentioned, is to put the system together. Putting the system together, and this is hard for neuroscientists, particularly those interested in higher cortical functions, to grasp, is more than just a neuron. Putting the system together means oligodendrocytes and astrocytes. So if one wants to use these, and this is really what you want to be doing, you're still, like I like to say, a translational developmental biologist, which means you need to understand first that when you do a transplant with a stem cell, there's a dynamic and both elements change, and they change in response to these various arrows. So I'm listing what has been the goal, kind of, the aims of the stem cell field since its inception. Notice that I'm listing it not chronologically, but in the order in which I think we will make an impact on healthcare. Way down here at the bottom, this is where we began, and it is still probably the bridge too far. And it's because we're still trying to figure out these arrows, and every time we think we understand one of the arrows, we realize it's wrong, and we got to go back to the drawing board. But in the course of doing that, we've hit a lot of these others, and this has become very, very intriguing, and this is what my hour is going to be about. Predicting, but it's not so much predicting. Actually, I wanted to have a whole alliteration with P's, which is why I did it. But it's more modeling a disease in a dish so that you can understand what's going on in a disease. Who's going to get it, how they're going to get it, what's the mechanism, what's the target, and how can you address it. So this concept, and this is probably starting to dominate the stem cell field more than any other aspect, is doing disease in a dish. Now what that means is taking a normal cell, or a neuron, and assaulting it in ways that you think a disease progresses. Now obviously, that is known for some diseases much more than for others. Or taking a cell from a patient that has a particular disease and having the hubris to believe that that cell that you took from the patient, particularly if you turn it back into a stem cell and make it a kind of particular nerve cell, has the disease in a dish that that patient had. In fact, I was warned at dinner to not go too heavily into saying that a single cell can have a disease. So I'm going to give that caveat. I get it. I know that. What I'm going to do is tell you how the stem cell field has tried to struggle through this, given that we work with cells. Now, once you have that cell with a disease, then what you hope to do is find a mechanism, a molecular profile, a fingerprint, a target, a prognostic, and a way of screening drugs to find a drug against that target. I'll be talking a lot about that. Now, obviously, the IPS field catapulted this area of research dramatically, but we always were thinking about this from the earliest days of the stem cell field. What we thought is that at post-mortem we could get brain cells, neural stem cells from adults or from miscarriages and fetuses, and we pull those out and make stem cells. And then with the advent of the embryonic stem cell field, in other words, from leftover blastocysts after IVF that were not implanted, we'd pull those out. And some of them, particularly if they were diagnosed by pre-implantation genetic diagnosis, you'd make an ES cell, you'd turn that into neural stem cell, and it would have the disease. All of these kind of depended on serendipity, chance, and something dying, which is certainly unpredictable, and you can't get all the controls you need. So what the IPS field did, this notion that you could take from a living patient, a very accessible cell like a skin cell or a blood cell, reprogram it back to being like it was in the embryo, like an epiblast, and then differentiate it, that opened up all kinds of possibilities. You could have large sample sets, you could have living patients, you could do clinical correlation. Now, what that also gave rise to was, well, this is now individual cells from individual patients. So it started increasing this notion of personalized medicine. Now, this is something that we're all thinking about. And as a pediatrician, and right now, we know the importance of personalized medicine, meaning kind of tailoring, figuring out what it is the disease you're going to get, and tailoring a therapy that's specific to your pharmacogenetics or whatever. It's hard to start getting together the database. And then I realized, as a pediatrician, in fact, as a neonatologist, I have a great opportunity to get everybody, wide capture, no cherry picking, in a prospective manner out of the starting gate. And that's because everybody's born with this. And what we have started doing, I don't have data for this, I can just tell you what we've launched. We can take the stump of the umbilical cord of all patients and just culture it. And we can do omics on it, turn it into iPS cells, which of course give rise to all the cells in the body, and most importantly, going forward, start doing clinical correlation, which is the key. So not only looking at the omics from a molecular point of view, but what I like to call the phenomics, what's going on, and constant correlation throughout the life. Which kid, in the newborn period maybe has hypotension or a cardiac problem. But then as you start going out, who develops diabetes, who develops a brain tumor, who develops autism, who develops a psychiatric disorder or learning disabilities or a degenerative disorder, and constantly doing that and putting together a database. So, but disease in the dish is kind of capturing the imagination of the field, and that's where I think we're going to make the greatest impact, and that's what I want to spend essentially the formal part of the grand rounds talking about. Now, what that says, and it's a conceit, and I recognize that, is that these cells, these are iPS-derived neurons, taken from skin, turned into iPS cells, and turned into neurons, that these have a disease. And let me at least tell you where we've taken this. And I completely understand that we always have to go back into the patient, but I'm going to try to make the argument that even in doing this, we've tried to do this kind of correlation all along. Now, it's not enough to say, well, I have these neurons now, and they have a disease, or I'm going to do something with them. The first challenge that we realized that we had, and I'm talking on behalf of the field. Let me also mention, I recognize that the audience is very diverse. Everybody has different interests, different backgrounds, has different focus. So I'm going to show, for those that want to know a lot of the details of the data, it will be up there. For those that want more the take-home message and the overview and the large story, that's what I'll try to do and capsulize as I talk about it. So if the details are not of interest, don't worry about it. It's more important what the major concept is. So the first thing is, what do you profile? Once you actually have these, remember I said the cells can give rise to everything. Well, what we actually realized is that even though we all thought, yeah, let's just have pure neurons in a dish, that's just not the way the body is put together. And David Cherish and I found, if you accept that the ES cell is a model of the epiblast, we found that straight out of the starting gate, the vascular system and the nervous system pattern each other in an obligate fashion straight away. And they do it through these various kinds of crosstalk that I illustrated over here. But if you get rid of one of them, everything falls apart. They must. Now, the interesting thing is, what leads the charge is the neural crest. It gives rise to the autonomic system, not the central nervous system, which is what every neuroscientist likes to focus on. After the pattern is formed, then the neural tube, the central nervous system, goes out and patterns that. But what that says is, already, we cannot focus on pure populations of cells. It's not going to give us the right story. It's not going to give us the right story on development. And as this paper illustrated when we were trying to look try to treat ALS, it doesn't even give you the insight into a disease. What we found was, for example, that when we went into an ALS mouse and used neural stem cells, we got great rescue of the animal. But it wasn't because we were replacing motor neurons. It was because actually we were, now what we learned was, and as the field of ALS started progressing, is we also learned that the disease, yeah, the motor neurons are diseased, but it's actually the environment that actually is equally as toxic to motor neurons. There are astrocytes that are not doing the protective job. In fact, they have toxic molecules against the motor neurons. And what we were able to do, not replace the motor neurons, but block this process, make the animal's own neural stem cells stop from spitting out toxic astrocytes, replace them with our own kindler, gentler astrocytes, and also protective gray matter oligodendrocytes. And that's why we're getting a rescue. So even in disease, it would have been wrong for us to say, we just want neurons. That's not the case. Now, you might say, well, how's that relevant to psychiatric disorders? Well, many of you are familiar with Steve Goldman's paper just about 6 months ago showing that he could recreate in an animal model through transplantation of glial cells a schizophrenic phenotype, at least in the animals. So what about, now that you figure out the cell, what's the right metric to look at? Now, most of us all start out wanting to look at the transcriptome. Now, everybody has a different take on how valuable that's been. Skeptics could say, we've had a decade of GWAS, and we're still in this kind of correlative association mode. We're starting to get there, but it hasn't, it didn't turn on the bright light that we hoped initially. So we start going up higher, looking at the epigenome. And as I'm going to talk about, looking at the proteome, what actually makes the cells work, and not just the proteome, but how the proteins get changed. And in fact, a study that we did when we looked at what changes as an undifferentiated cell becomes a neuron, what was most dramatic and changed the most in this spectrum of differentiation from undifferentiated to differentiated was not just proteins, but it was actually what the phosphorylation state of the proteins. So we're not talking about a gene, we're not talking about a protein, we're talking about how that protein gets modified. And when we actually try to do a correlation in this differentiation study between mRNA and the phosphoprotein that made the cell work, we did this kind of plot, and it's a mess. And that's the point. It is a mess. It was only about a 50% correlation. What really determined what the cell was doing was the phosphoprotein. Now, of course, we're starting to go higher. Some people in this department are doing this, looking at the metabolome, looking at the connectome, and now putting it together in 3 dimensions, multiple cell type, into the organoid. So Ilya Singech in the lab started recapitulating this process by making mini neural tubes. And Lena Mastrangelo decided she wanted to try to start modeling these migrational defects. Who knows whether we can actually do that or not, but compared to normal brains, this lissencephalic brain and iPS cells derived from that, particularly when LIS1 was inactivated, she started seeing this kind of very interesting scenario. Cerebroids that were kind of normal, and then cerebroids that almost looked as if they had, the organoids almost had the smooth brain, which is the appellation which is given to kids with lissencephaly. So the next thing, and this, now we're getting to the heart of what is the most difficult in this entire field, and is how do you start assaying a clinically meaningful predictor? Now, what was easiest in this field was to simply jump to a monogenic disease, because at least you know the gene, you knew probably what the gene did, you knew the pathway, maybe the cell type, and you could start getting to mechanism. So the field began looking at monogenic diseases, and we were no different than anybody else in this field. We decided, for example, to model Parkinson's disease, but not sporadic Parkinson's disease, familial Parkinson's disease, which is the minority of Parkinson's disease, but at least you have a gene. And we were looking at the LRRK2 mutation. So Lena made these cells, these are neurons, with the LRRK2 mutation. And we were now able to go off to the races. This is not a Parkinson's talk, so I don't want to spend time going through all the details here. I just want to show you what having a gene and iPS cells with that gene defect can allow you to do, to make an enormous amount of quick process. We started entertaining this notion that what's going wrong is really a lysosomal opathy. And the lysosomes are dysfunctional because autophagy is dysfunctional. So now we're able to start doing, straight away, start doing assays, the various kinds of autophagy. So this is called chaperone-mediated autophagy. It's based on a receptor, and you just look for whether this red dye starts disappearing. And the quicker it does it, the better the autophagy. And sure enough, we found that the LRRK2 mutation was really impaired in that. But the PARK1 mutation actually even worse. Oh, so that was kind of interesting. And then there's another kind of autophagy called macroautophagy. And that's when the autophagosome and the lysosome merge, and then whatever has to be degraded gets degraded. And you can start looking at changes in colors. And there we found, well, LRRK2, that's really impaired there. PARK1 mutation, not so much. So we started being able to put together a little bit of story of what was going on. Now, the key to all of this, and again, this is what happens when you can model a monogenic disease, is you can do proper gain of function and loss of function. Genome editing, now we could correct the LRRK2 mutation, knock in the LRRK2 mutation, do double mutations, and start really finding what was going on in this process, and finding actually with a double mutation, it looked like these two familiar forms of Parkinson's disease have defects in different regions of the whole pathway. And they interact like this, they become additive, and they compensate for each other, and we could put together a whole scenario. And not only that, we could actually have specific inhibitors of LRRK2 and of lysosomal function and things like that, and watch the aggregate starting to form. That was all because, and we have no idea whether this is what happens in sporadic Parkinson's, but at least we had a handle and we knew this is what was going on in familial Parkinson's disease. That's what you can do when you have a gene with this kind of technology. And now we're getting to the meat of the grand rounds. I know you're going to say that I wasted the first half hour, but I think you needed this background to understand what I think, how gratifying this was to be able to use iPS cells to start tackling this problem. So, like I said, almost everything in the iPS field has been with monogenic diseases. When you start tackling these more complex polygenic multifactorial diseases, the best we've been up until recently is being descriptive, kind of describing the phenomenon, phenomenological, descriptive, correlative. But I became intrigued, and because even though I'm not a card-carrying psychiatrist, I was trained in psychiatry in getting my boards, that these were really the poster child for these really, really tough diseases. So we realized not only would trying to attack these diseases be important for the diseases themselves, but it could provide a roadmap for our entire field as to how to start attacking these kind of diseases. And I have to say, most neurologic diseases, certainly most pediatric diseases, are not ones that we completely understand. So what Brian and I came up with was something that we called the molecular can opener strategy. And this is going to tell you the whole grand round. So if you see no other slide, you'll come away knowing what we found. But I'll start that, and then I'll piece it together and kind of unpack how we came to this. The notion was, and Brian kind of enlisted all these other people that were in the lab that contributed. What the can, of course, is these complex diseases that we don't know anything about. We know the phenotype, but that's about it. We don't know anything else about it. But we got to get into the can. And what we realized was that of all the psychiatric disorders, there was one condition that actually could give us a can opener. And that was bipolar disease that was responsive to lithium. Because after more than 100 years of lithium being used, we still don't really know why it works in this kind of disorder. And I don't have to tell this audience just what a devastating and prevalent disease this is. Well, the notion was that if we could find lithium's target, protein target, that would give us a pathway we call the lithium response pathway. We could map upstream and downstream, and if lithium reverses it, this pathway must be essential somehow to the pathogenesis of this disease. And what we wound up finding is that the target was a molecule I'll talk a lot about called CRIM2, which is pivotal in cytoskeletal regulation, which is necessary for many kinds of things, and I'll allude to that. Now, again, it's not that we didn't understand what lithium did. That wasn't the problem. Lithium did too much. Lithium did everything. And it did it not just in vertebrates, and not just in mammals, and not just in primates, but in invertebrates, and flies, and worms. So that was not helpful. What these guys did, led by Brian, was to take skin biopsies from bipolar patients, lithium responsive, non-lithium responsive, a whole host of patients, turn them into iPS cells, then turn them into neurons, and then do actually a fairly simple proteomic trick. And it's probably the simplest proteomic maneuver. On the other hand, it also is exceedingly stringent. So that's where Brian started out. And first thing I should just mention is that we used, and I won't go into the details except simply to say that we used a recipe that we had devised that can go from pluripotent cells, which represent the epiblast, in a step-by-step manner to take you to neuroectoderm. And the neuroectoderm would give rise to neurons that were dorsal, cortical, GABAergic, or glutamatergic interneurons. Now, what was interesting is straight away we saw that disease, iPS cells from bipolar patients and from normal patients, or any psychiatric patient that we looked at, did not have a defect in differentiation. We got the same number of glutamatergic and GABAergic interneurons. So that was not where the defect was. And these were, true electrophysiologically good interneurons. So now we have the neurons. And the proteomic trick is called 2D Dodge. And what you do is you just take the same clone, you expose part of it to lithium, one split to lithium, the other split not to lithium, and then you do something that separates them based on change in abundance, charge, and molecular weight. So what you're looking for are things that are not yellow, things that have changed, either pure red or pure green, and then you just pray there's not too many spots, not too many differences. And we lucked out that there were only about 15 spots in this experiment that we could pull out, do mass spec, and identify. And then you take that and you do bioinformatics. And what popped out as central to the whole thing was axonal guidance signaling. And what was pivotal to that was a central molecule called collapsing response mediator protein 2, or CRIM2, a cytoskeletal modulator, which is important for axonal guidance and channel formation and dendrite formation and synaptogenesis. Interestingly, it's located on a part of the chromosome that is very familiar to this audience. It's been heavily associated with neuropsychiatric disorders. And I'm not going to go through the things that it does except to say that it is pivotal for development. Now, many of you know its more famous cousin, semaphorin 3A. Collapsin is the old name for semaphorin 3A, which everybody knows because you need that for growth cone growth and growth cone collapse. But what was cool about CRIM2A was it sort of gave us this developmental field that we know exists in bipolar disease, in many psychiatric disorders. It also gave us a sense of where some of the inheritance could come. Its regulation is completely pivotal. It pivots on phosphorylation, and I'll talk a lot about that. And it's susceptible to external cues. going to skip this except to say that Mike McCarthy helped us with a whole lot of other bioinformatics. So where is CRIMP2 being expressed? Well, it doesn't start coming on until you start neuralizing the iPS cells, and heavily when they become neurons. Where is it located? Well, we found it's located in all the regions that you might expect. You might expect a neuropsychiatric disorder to be represented. Heavily in the hippocampus, even in the cerebellum, in the striatum. And now I'm going to show you, we didn't discover CRIMP2, and some of its regulation was known. Let me show you what is known about its regulation, and then I'll show you how we superimpose our knowledge, particularly relevant to CRIMP2, to bipolar onto that. So what CRIMP2 does, it binds cytoskeletal elements. When it's active, it binds, and then you get outgrowth, or you get activity. Now, it gets inactivated when it gets phosphate groups placed on it. And it starts off with CDK5, puts the first phosphate group on, which primes it, and then allows GSK3 beta to put on 2 additional phosphate groups, and it releases the cytoskeleton. So this is the inactive form. Now I should say, this is a normal function in all of us. Toggling back and forth between active, inactive, binding cytoskeleton, releasing cytoskeleton is normal. It's a normal adaptive mechanism. But now we needed to know, how did lithium really superimpose upon that, which means we had to start mapping the lithium response pathway. Well, how do you do that? I'll just give you one example of how we started doing that. First we found, interesting, lithium doesn't change total CRIMP2 at all. So the total protein is unchanged. So that starts becoming interesting. But we found through the bioinformatics that the lithium response pathway, or the CRIMP2 pathway, completely superimposed, or heavily superimposed, on the GSK3 beta response pathway, not unsurprisingly. So that's where we started. So we found that lithium did interact directly with GSK3 beta. And if that's the case, if that really is completely downstream, then a direct GSK3 beta inhibitor should do exactly what lithium does, at least with regard to CRIMP2, and that's exactly what happens. Lithium decreases the phosphoform, so does a direct chemical GSK3 beta do it, and it does it in a dose-response manner. Total CRIMP2 not affected at all. So, and again, here we show that lithium now increases inactive GSK3 beta, and that's illustrated over here, lowers phosphocrimp2, in other words, lowers the inactive form, as illustrated over here. And if you start putting it together, whether you use lithium or a chemical inhibitor of GSK3 beta, what starts to happen is you lower this, and once you lower the inactive form, now you start getting rebinding in these pull downs, rebinding of CRIMP2 with cytoskeleton. So things started looking pretty good. It started falling into place. And we just went through that with every node in this entire pathway, showing that lithium does interact with the GSK3 beta pathway. So GSK3 beta dependent pathway, parts of the pathway, and also GSK3 beta independent parts of the pathway. So that became important. But what we started recognizing was, I have this, you're not seeing that, are you? I'm getting a memo that I'm supposed to start lab meeting. So the phosphorylation state started becoming important for how this molecule worked. Now, what was it doing? So we started looking at the neuron to see what was the respective distribution of phosphocrimp, inactive crimp, versus active crimp. We started looking at one of the business ends of the neuron, and that's not only on the dendrites, but the dendritic spines. And what we found is CRIMP2 was expressed all throughout the neuron, but when it was active, it was in the spines. When it was inactive, it was out of the spines. And it would go back and forth based on its activity state, as you can see. So now we started getting the sense that, okay, the level of activity of CRIMP2 seems to be pivotal on how or among other things, how dendritic spines are active, inactive, forming or not. So now what you do is you just do a blunt instrument. Just knock it out. Knock CRIMP2 out. And sure enough, what you see, you knock out CRIMP2 entirely, and you get completely aberrant dendritogenesis and dendritic formation. And you even get abnormal branching of the dendrites that just branch too soon. It's called a proximal dendrite abnormality. And very key, what you start seeing are diminished dendritic spine densities. So this becomes actually pretty important, and that's simply qualified over here. Now, as I mentioned, that the amount of total crimp was not different between the neurons from a bipolar patient and a normal patient. What was different is this, and let me just run you through this because this started becoming interesting. We're staining now for phosphocrimp2. And as you can see, these are normal neurons, these are bipolar neurons. And it already starts looking, and you can quantify this by doing pixel counts, and Steve Haggerty, who was our collaborator, did that. This looks brighter, and sure enough, there's more phosphocrimp2. Now you add lithium, and you get rid of the phosphocrimp2. Now, if you start quantifying this, it starts becoming kind of interesting. So lithium drops the phosphocrimp2 and the bipolar neurons from here to here. Well, it's interesting. This drops it to a level that already is the starting level of a normal neuron. And of course, so what we started wondering, they started entertaining this hypothesis that maybe the set point of inactive to active CRIMP2, it's not the total, it's the ratio, the set point, is abnormal, is higher in these bipolar patients. And then if you buy this, here's another way of looking at it, the CRIMP2 binds the cytoskeletal elements. It gets phosphorylated, this is kind of normal, and when it gets phosphorylated, it releases those, and you get neurite retraction. But what happens is the ratio is just too high, the set point, the thermostat's too high, and what lithium does is just normalize it to a normal level. And sure enough, so I just showed you single neurons, but now we started doing Westerns on just many, many, many patients. And what you saw, in fact, is in the lithium response bipolar patients, sure enough, their ratio was too high. It was different than normal patients. It was even different than lithium non-responsive bipolar patients. But what lithium does is now lower it to a level that's the same as unaffected and even the same as patients with that are lithium unresponsive. So now the next thing you do, well, this is just cells in a dish. So you actually go to a real brain bank, go to actual brains of bipolar patients. And what becomes interesting, in fact, is the bipolar patients have an abnormal ratio, but also their spines are diminished. They look just like the mice in terms of their dendritic spine. And the patients that had this also had that. And this is illustrated here. We looked at a number of brain banks. We did the McLean's Brain Bank, the Pittsburgh Brain Bank, and in fact found the ratios were abnormal in these patients that also had abnormal dendritic length, meaning they branched too soon, and also diminished dendritic spines. However, starting lithium had a high cost. It normalized the ratio and normalized the spine density. So now we're talking about either cells in a dish, or histology, or mice, or postmortem specimens. But what about the actual activity? For that, you actually have to go to living human neurons in a dish. And Alicia did this, and she found that the neurons from the lithium-responsive bipolar patients actually had a very quick influx and efflux, a very steep slope, very different than normal, different than lithium non-responsive patients. But what lithium did was normalize the slope. So now we had some functional correlate for what we were saying before. Now, interestingly, in a mouse where you knock out the target of lithium, a CRIMP2 knockout, lithium doesn't change this at all, which is what you would expect. Lithium also increases spine density and spine volume. In normal animals, it doesn't do it. In the animals are missing the presumptive target of lithium. Doesn't happen at all over here. So now we're starting to work with this hypothesis that CRIMP2 seems to be fairly pivotal in dendritic morphology, which leads to function, which is important for networks and things of that sort. And then, of course, the prediction would be, and that if the ratio is wrong, you just get abnormal network formation. And of course, what Cameron Pernia did was show that, in fact, downstream of CRIMP2, things that would go along with that hypothesis are true. So cofilin is abnormal, and tubulin is abnormal. It's too low if it's knocked out, but in a dose-response fashion, and I'll get into this, how you can do a gain of function of CRIMP2, you get even more activity like that. And when you do the bioinformatics, as you might expect, calcium signaling is immediately, prominently downstream of CRIMP2. Now, was there disease specificity? Well, kind of interestingly, and I'm going to start going a little more quickly now, yeah, in lithium-responsive bipolar disease, it was too high. Lithium lowers it to a level of unaffected. Major depression patients, that's not abnormal at all. If you go and simply look at GSK3 beta, lithium-responsive bipolar patients, overly high in their phospho content, but this lowers them back to baseline, almost in a dose-response fashion. So there seemed to be some disease specificity. Now, it's interestingly, major depression does not have an abnormality in CRIMP2, either total or in its ratios. Neither did other neurologic diseases we looked at, not Parkinson's or other things like that. Was there drug specificity? Well, playing with CRIMP2, of the ones we looked at, and Brian looked at a bunch of them, I'm only representing a subset here, only lithium did that. So we started thinking maybe we now have a nice biomarker for this. Now, reviewers used to say to us, well, maybe this is just GSK3 beta is much more famous. Maybe this is just a GSK3 beta abnormality. Well, it wasn't, because it wasn't just a diminished level of inactivated GSK3 beta. That wasn't different across the patients. This really was true to this particular molecule. But then the next thing you have to do, and I know this is the third rail in trying to model psychiatric disorders, is we knew we had to go to some animal model, and what we did was embrace the one that thanks to John's work, probably is the most reliable and the most famous model of lithium-responsive bipolar disease. We decided to look at animals exposed to methamphetamine to see what would happen. So we started with mice, and sure enough, lithium given to the mice actually increases inactive GSK3 beta. It also actually decreases inactive phosphocrimp2. Now, this started becoming interesting. Methamphetamine increases, not only in the dish does it increase inactive CRIMP2, but also in the animal. It doesn't change total, but it does change the level of phosphocrimp2. So we already had a feeling this is going to be kind of interesting. So we did this kind of assay that I know you all know really well. How much is quantifying how much time does the animal stay in the unprotected center, normally and then after giving methamphetamine. But we decided to do it in a very informative molecular fashion. We decided that we would do it, and this is obviously an animal, wild-type animal who's been given methamphetamine, who spends very little time in the center, mostly circling the periphery fairly manically. We decided that if we're really going to nail down where we think lithium's site of action is, we need to do a molecular manipulation that actually changes the precise residue, the precise motif where we think lithium is working. So now you take a mouse, and remember I told you the whole thing, how you can prevent this animal from being phosphorylated is you just get rid of the primer, and the animal becomes a non-phosphorylatable animal, and that's exactly what we think lithium's doing. That's the site of lithium action, at least hypothetically, or at least that's our hypothesis. And again, You take this mouse, and it's called the knock-in mouse. And remember, it can't get inactivated. It has hyperactive constitutive CRIM2. Already, we're starting to get a little bit interesting. Look at this. In response to methamphetamine, as opposed to into wild-type animal, you don't get phosphocrim2 increased the way I showed you before. So now you take the animal, you take your non-phosphorylatable animal, and you put him in there. You give him methamphetamine, you have the wild-type animal. And of course, the wild-type animal really stops spending time in the center, starts wildly circling the periphery. But the non-phosphorylatable animal, he just keeps smoking and joking. It's like he's never seen the methamphetamine. It's as if he's on chronic lithium. Now, I'm oversimplifying this, but I'm just a dumb neuroscientist, so you have to, and pediatrician. But it looked as if we were able to emulate with a molecular manipulation precisely the site on the molecule and the motif on that molecule that we think lithium's working. What that told us was that to start getting into some mechanism, you can't just do iPS cells. You really need the mouse models and the human material. But what started becoming interesting, let me just, could I just take 15, would it be okay if I took another 15 minutes? Because we got started late because of that. Okay. I'll finish up, and I want to start getting into some. So now we had some tools, however. Remember, we have the knockout mouse, we have the wild-type mouse, and we have an animal, a knock-in mouse, who has constitutively overactive CRIM2. So this is a great tool to start even better dissecting the mechanism of what one thinks is going on. So what Cameron started doing is taking these mice, and we'll go back to humans. But the mice, gave us a very manageable tool. The more inactive CRIM2 is, the more excitable, or let's say this, the more calcium events, the greater the kinetics of calcium in those kind of cells. And that's what this kind of shows over here. So now we have very hyper-excitable cells. Now this is in the mouse, but Cameron was able to show that exactly the same thing happened to the human neurons, the human bipolar neurons. And just as we showed you before, lithium normalized that degree of hyperexcitability. So now we started going, well, we're talking about networks. We're talking about how networks could be perverted, but can that give us some insight into really how these networks may malfunction? And we decided that what we would do is say, well, conventional wisdom, maybe not to psychiatrists, but to the lay public, is if you have hyperexcitable neurons, then your network should be hyperexcitable. They should be just quicker than what a normal network is. And maybe that gives you some of the phenotype. And again, these are people that have a narrow view of what bipolar disease is. They don't see it as a cognitive problem or something like that. Well, we decided to see whether that was really the case. So we started doing something called microelectrode array. And that basically is you take all the neurons, you stick them in many, many, many wells, and underneath there's a way of looking at how they fire and how they start forming networks together. So you're able to extract data like this. You're able to start seeing the neurons firing, and then what the computer actually does is give you spikes and shows you who's connected to whom and what kind of networks you see. Well, what Cameron actually found is, here's normal networks, normal neurons. Here's the networks that you see from at least the mouse model that has, with a little bit of conceit, we're saying could model some aspects of lithium-responsive bipolar disease in this CRIM2 knockout animal. And sure enough, what you see is decreased signal complexity. You see decreased network burst duration, decreased burst frequency, decreased mean firing rate. So what we're actually seeing are impoverished circuits, even though the neurons themselves seem to be a little hyperexcitable. Now, why might that be the case? Well, one simple explanation could simply be, well, you just have fewer spines. That's no surprise there. And That's not so interesting. Or you could say, well, you know, you just told us that CRIM2 affects this calcium channel, and what simply happens is, as you inactivate CRIM2, you just have less signaling. So that's not all that interesting. But what did start becoming interesting is, hopefully this is going to play, is what we started seeing over here. All right, I'm not sure if you can see it, but notice this is calcium lighting up. Now notice it's not a ripple effect. It's not calcium rippling across the field. Everything is lighting up simultaneously. So this is something that many in the audience, particularly those who deal with epilepsy patients, would know. This is called synchrony. So this is the way normal cells should be firing. And this is the way the wild-type neurons fired. It was just kind of random rippling across. But here's what, oops, sorry. Here's what happens when you have excessive synchrony. And that's what the bipolar neurons were doing. And Cameron, sorry about this. Computers decided it was. Cameron was very proud that he came up with a whole formula to be able to quantify that. And what he found was that with decreased CRIMP2 activity, which presumably maybe emulates some aspect of lithium-responsive bipolar disease, there was increased synchrony, as you can see over here. And what he saw, however, interestingly, what he saw in the mice was exactly what he saw in the human bipolar neurons over here. So counterintuitively, the more excitable a neuron may actually make for less effective neural connectivity, neural circuitry, and neural networks. And for other physicians in the audience, I like to equate it to the cardiac system. We all know that to increase your cardiac output, you become tachycardic to an extreme, to an extent. You know, if you increase your heart rate, you increase cardiac output. But if you increase become too tachycardic, SVT, you actually lose cardiac output. So we started using this cardiac analogy to try to understand what was going on. What it did tell us, however, was that we in regenerative medicine have probably been getting this wrong for a very long time. What we need to think about in terms of diseases is not neurodegeneration, but networkopathies. And that we've always been focusing, so I'm getting back to kind of my roots, that what we've been seeing, what we see in these diseases is not necessarily neurons falling out, and improving neurogenesis or putting back neurons is probably not what we need to be doing. What we need to be doing is thinking about protecting and restoring networks. Now, that was one important insight. But remember I said one of the values of being able to do this is to start now seeing whether you can get drugs that can modify this molecular pathway. Well, that's one of the selling points of iPS cells. So now, could we find a better lithium? Now that we knew really what lithium was doing. With the lithium response pathway, we had all kinds of novel ways of what the goal would be, screening for something that lowers phospho, lowers inactive CRIMP2, that'll make spines and networks work better. So we're very blessed at having probably one of the best drug discovery infrastructures in academia at the Sanford Burnham. Also, we teamed up with Steve Haggerty, who at the Broad also has a great discovery infrastructure. And what you can do is just screen thousands and thousands and thousands of drugs, including FDA drugs, FDA-approved drugs that can be repurposed. And what this simply shows is what lowers phosphocrim2, and at least give that a shot. Now, this is very preliminary data that Brian Tobey did based on some of the candidates that Steve Haggerty came up with. One thing is, would any drug decrease the methamphetamine effect? So again, same thing that John discovered and that you're very familiar with, animal in the open field, and in the center, and then you give, you give methamphetamine. The wild-type animal no longer spends time, in the center, madly out in the periphery. But this drug, interestingly, huperzine, and we can talk about all the other things we have to look at in here, but intriguingly, huperzine did exactly what lithium did in there, and that was discovered because we did a screen for lowering phosphocrimp-2. So we're getting encouraged that maybe we can find something that is very specific working on the lithium response pathway for this particular disease. Now there's something interesting, and I'm going to introduce this, I just have suggestive data, and I'd be interested in getting feedback on this. So I gave you this scenario already. What happens is when you have phosphocrimp-2, inactivated phosphocrimp-2, these are released. So we started wondering, what happens to that tau when it's released, and particularly what happens to the tau if it's excessively released and released inappropriately? Well, we're all familiar with tauopathies, and the most famous tauopathy is Alzheimer's disease. Sure enough, what our collaborator Yoshia Goshima found was in the Alzheimer's plaques, there's excessive phosphocrimp-2. And what we're able to find in looking at these bipolar neurons is that there was an upregulation of APP and increased constitutively, when you constitutively increased CRIMP2, in other words, made it more active, decreased phosphocrimp-2, you would actually also see a decrease in the level of APP and tau, and importantly, phosphotau. And lithium also, in these neurons, not only prevented the dimerization or decreased the dimerization, see a greater shift, but decreased the level of phosphocrimp-2. Now, this audience knows, this is old literature that I know everybody here knows. But with these observations, it made us start just looking. Brian started looking back at some of the old literature and started finding, in fact, there has been in the older literature a report of bipolar patients being more susceptible to some dementing diseases, but those that would take the lithium actually less. We've become intrigued with chronic traumatic encephalopathy, an acquired tauopathy based on repetitive concussive injuries. Well, this is tau, but if it's true, and remember I said this toggling back and forth between phosphocrimp-2 and regular crimp-2 is a normal adaptive mechanism in all of us. When you get hit on the head, it spikes to prevent inappropriate sprouting in the midst of inflammation, and then it goes back. What about if it never goes back, or it just gets repetitively spikes and spikes and spikes? Could that in fact be what seems to be going on here? Could the same lithium response pathway be operative in this kind of disease? And we know lithium is useful in head injury. That's an old literature. So I'm starting to wind up now, but I've completely embraced not only how important this field is for the patients themselves, but I really believe that this can give astounding insight into a whole range of diseases, and certainly can inform a regenerative medicine doc like me as to what we really should be doing. And I honestly think that we've been getting things wrong for 20 years, or at least we need to modify it a little bit. I think it's kind of cool that accessible tissues like blood and skin can ultimately give you insight into something as inaccessible and complex as neurons and neural circuitry in the brain. That I find mind-blowing. But if you have that, could this be some way, some biomarker to help guide clinical trials, to help with diagnosis of patients, to help follow and monitor their progression on various drugs? So I'm intrigued with trying to explore this a little bit more. Now, as Brian, I'm sure, can tell you, some of the first stuff we did publish last May. And as you can see, there were 51 authors. And I think, as Brian may have confided in you, trying to get this out was almost akin to the Normandy invasion. but we did it. But what it also says is that really good science involves many, many disciplines of people working together. And the 51 authors represented 10 different disciplines, multiple institutions, 5 different countries. It involved chemists and biologists and animal modelists and psychiatrists and neuroscientists. And that really is the way I think we have to do big science from now on. So I'm going to end here with this with the summary that, we think that this lithium response pathway, which really pivots on cytoskeletal modulation, plays an important part in bipolar pathogenesis. It also tells us that, most of us went into neuroscience because we loved higher cortical function, language, and complex behavior. And for me, it was humbling to realize that all of that could come down to something as basic as a cytoskeleton. We also recognize that as well as transcriptomics, proteomics is important because what we're talking about is a disease. The gene's not abnormal, the protein's not abnormal. What was abnormal was the regulation of the gene product after translation and after transcription. That this ratio could be an important diagnostic, that I'd love other people to use the molecular can opener approach. Interestingly, what this is kind of a reverse drug discovery. So I just want to plant the seed. By reverse drug discovery, most of the time, you find a mechanism, and then you search for a drug that changes that mechanism. This was the reverse. We had a drug. We just didn't know how it worked. So we went back, found the mechanism, and now we can find a better drug. And I think we all would like to do that. And as I mentioned, I think for more broadly, it teaches us a little bit about, not to just jump to conclusions. Hyperactive neurons could actually mean hypofunctional neural networks, and that, as I mentioned before, regenerative medicine, I think, needs to start focusing on networkopathies. Now, since we published this, I think it's very refreshing. Dan Geschwind, I think, as you know, I think has made a really important contribution to the transcriptomics of these diseases. Now, as you know, the limitation of transcriptomics is it's still descriptive, it's not mechanistic, it's still correlative, but it does tell you whether you're on the right path. And remember I said that we found neural networks and bipolar disease to be much more associated, CRIP2 to be much more associated with neural networkopathies and things like schizophrenia and degeneration, and that major depression really had nothing to do with this pathway. And interestingly, that's exactly what Dan found in the transcriptomics. A huge association between schizophrenia and bipolar disease, and actually not so much with major depression. So with this, I'm sure I've said things that hopefully provocative, hopefully won't kill me too much, but I'm willing to entertain some questions. I recognize that this field makes for absolute Talmudic debate. I know this is me. Hopefully this will be not too many of you out there, but I'm willing to entertain any questions, and I particularly want feedback from an audience as knowledgeable as this. So thank you very much.
Well, wow, what a great journey you've put us into. So thank you. Now, immediately we have John Kelso. Just speak.
Evan, that was a fantastic talk. I mean, I'm just really impressed with the multi-levels and really taking it from top to bottom. It's a really cool discovery. Do you think your phosphocrimp ratio has anything to do with hyperexcitability, as Rusty?
Well, Rusty obviously wasn't looking at CRIMP2. He was looking at these populations, but I think it does, and I think we can explain why now too. I think what it's doing, it's regulating some of the spines, which then, of course, determines the degree of synaptogenesis. And also, cytoskeleton determines what a channel looks like. And I think that it's affecting the calcium channel. So I think the answer is yes. And importantly, I think that these kind of going back and forth, and this happens to all of us all the time. It's just, hopefully most of us it's regulated for most of us. So the answer is yes.
And just one comment in defense of GWAS. Some of our first hits were cytoskeletal.
Oh, you're finding cytoskeletal.
Yeah, ANC3,
Yeah.
SIN1, a couple of different things that are cytoskeletal elements that were a surprise to those of us in that field.
No, I know. And so now, when this came out, I started going back to that and going, what do those guys find? And, and you're right. You're right.
Really, I agree with John. It's a real tour de force. Very, very impressive. It's striking to me that that really kind of central to this whole result is a spot on a 2DG gel of what's different in the cells in the presence of lithium versus no lithium. What about the other 14 spots on the gel?
Well, so let me emphasize that was just the key. We would, if we had just stopped and said, well, we did this, here's the spot, here's this, we wouldn't have a story. It would be purely, it was, that just kind of shined a light at that. And then we had to do all this other stuff.
Well,
now, when we did the bioinformatics, so what we did do is start doing bioinformatics on all the other spots, particularly pathway, bioinformatic pathway analysis. And what started happening is all the other spots started, how you make these, dendrograms and things like that. They all focused on axonal guidance, all of them. And at the heart of the axonal guidance was this molecule. So CRIMP2 was at the center of all the other 14. You know what I forgot to mention, though? I didn't mention this. When we got this finding, and this is actually critical because we did one technique using 2D DIGE. We were funded by NIMH to do this study. We had to have a come to Jesus meeting at NIMH where we told them what we did with the money. Steve Haggerty also was funded to try to study bipolar disease. He was doing SILAC. And this is a true story. Brian Toby was at DC sitting next to, and I was sitting next to Brian, and he, by chance, was sitting next to Steve Haggerty. Brian went up to talk first and show some of the early data. And as Brian was coming back from the podium, Steve leaned over to me. Completely different technique, SILAC. He said, that's what we found too. So, when you have two completely separate groups looking at two completely groups of cells, doing entirely different proteomic techniques, and you come up with the same answer, I have to say that also helped a lot.
Thank you so much. I actually appreciated the half-hour intro because I think that it is complex and all the different considerations in this field are important for the consumer to know about, and especially somebody trying to do that kind of work. So one of the things you mentioned was picking the cell type, but most of your lithium and bipolar story was about neurons. Yeah. What about the astrocytes and the microglia? Because again, the GWAS studies and other things point to these immune mechanisms that might be mediated by some of these other types of cells in the brain. So any suggestion that the CRIMP2 story extends beyond neurons?
So CRIMP2 is actually very particular to neurons. And I actually think, now again, I'm really going off script and I'm just really spitballing here a little bit. But what I think is that lithium-responsive bipolar disease really is this dendritic neuronal kind of problem I should also mention, I don't think I emphasized, I actually, and I think some other people in the audience agree, I think lithium non-responsive bipolar disease is an entirely different disease. I think lithium-responsive bipolar is this particular disease, pivots on this mechanism that is very neuronal. And I think lithium non-responsive bipolar disease, not only is it a separate disease, it's probably not one disease. It's probably a group of diseases that phenocopy this, but each have their own different kind of mechanism. But it's kind of interesting. CRIMP2 is also abnormal in schizophrenia. What's not abnormal is the ratio. We've not published this, but we've been starting to look at this with Glenn Klenofsky. But it's just the total amount, and it's only the ratio. In bipolar disease, it's only the ratio that's abnormal. But there, I think in schizophrenia, I think that's not a neuronal disease primarily. I think that's why Steve Goldman can see these abnormalities in the glial cells. I think why there is such an association with complement and microglia. So I think you may even get networks that look equally abnormal. You may even get spines that seem, and synapses that seem to be equally abnormal. But I think lithium-responsive bipolar disease came at through this route, and that maybe schizophrenia comes at it exactly the way you're describing. All these other things that are also critical for molding the synapse. But that's purely speculative at this point.
Fascinating work. I was curious, in contrast to lithium, estrogen enhances dendritic spines in hippocampal CA1 neurons. It enhances neuronal synchrony.
Right.
It can potentially increase mania and rapid cycling and enhance cognition and memory. So I'm wondering, any thoughts about how that might work?
No. I mean, I'd have,
All right.
other than, that is the kind of thing that you just go back to the bench and you start trying to see how estrogen would map onto there. And, it could be something, it changes a kinase, which then changes phosphorylation of a whole slew of molecules, including this one. Estrogen's a perfect example of so many pleiotropic effects. And what our goal, I think, is to try to take something that has so many pleiotropic effects and at least try to drill down into how it does it. So I don't know, but I think it's important to study.
And the other thing, the conceptual model you showed about, the astrocytes and the Schwann cells and sort of the therapeutic milieu sort of took me back to my Psychology 101 class where, remember the baby monkeys, if they're separated from their mother, it causes lifelong trauma.
Yeah, the Harlow study.
Yes, right.
Yeah.
And if they put it with a cloth monkey,
Right.
then it prevents that.
Right.
And it reverses the hypercortisolemia, so you restore the diurnal pattern of cortisol.
Right.
So that's
right.
sort of a backward approach, but I'm just wondering, is that going to enhance phosphorylation? Or
It could. I mean, I think now, when you find an interesting finding, the danger for all of us, particularly for cell or molecular biologists, is to think that it explains everything. I think probably now it would be interesting to see how these various effects kind of come down to something as fundamental as a post-translational modification. So this would be the first disease, certainly I think the first in neuroscience or neuropsychiatry, that's a disease of a post-translational modification. I guess you could say some of the neurodegenerations if you buy ubiquination and things like that. But what I'm starting to find is that there's nothing new that we'll discover that people didn't find 40, 50, 60 years ago. And you alluded to some of it. We just have to figure out how it maps onto this.
Yes. Thank you for this very elegant presentation. And I'm interested in schizophrenia. And we have a research scientist working at the Gage Lab, essentially to tackle the astrocytes and its relation with a neuron and with various kind of culture to see in what way astrocyte is related to schizophrenia. And so that is all. In the future, are you getting into schizophrenia area from your talk, apparently from presumed non-responders, which may be another way of your future work, I guess.
Yeah. No, I'm very interested now in seeing how this would interact, particularly since Dan, we would have predicted that there would have been this overlap between schizophrenia and bipolar. Dan now kind of nails it. So of course I'm very interested in that. And I think your approach of looking at what the glial cells do to the neurons is exactly where the field needs to go.
Well, thank you very much. One final comment. So would one of the lessons, and I'll ask John as well, be that we should now really start everybody on lithium first, and if they don't respond, then they can go to those other things. Well, seriously, I mean, we're starting with all the other stuff and mucking around, but what do you think? Is that a reductio ad absurdum?
No, I have to say, in my training, I mean, and this is on neurology, just doing a psychiatric rotation. That's
neurology is okay.
What my teaching is that if it looks as if you have a patient with a particular constellation of symptoms that would make you think he's bipolar, start them on lithium and see whether or not they respond or not. So that was my teaching.
That's not what we do, though, is it? Yeah. Anyway, so it's some food for thought. I mean, here's some research. Anyway, thank you so much for a great talk. And we have a small tradition here in psychiatry that since you do claim some relevance to psychiatry,
I do.
That we now provide you with this very nice hat, UCSD Department of Psychiatry,
Great.
School of Medicine. And hopefully it will be also a homing device so you can find your way around the campus. You said you had some problems there. But if you wear that, people come up to you and say, oh, you're looking for psychiatry? There it is. So, thank you so much.
Thank you. Thanks so much for inviting me.
You can adjust the size.
Yeah, thank you.
Clean up your stuff, the cups you left under your chairs, take them to the trash. This is your Mr. Mom. Take your trash out. Huh? Believe me. Otherwise, poor Felicia and me have to clean it up.